The human heart has essentially 4 chambers, 2 ventricles and 2 atria. In the case of zebrafish, this system is slightly simpler, and we only have essentially 1 ventricle and 1 atria. I think that zebrafish has a major advantage that it is transparent during early stages of development. It allows us to do in vivo imaging of multiple organs during the development, but it also has the ability to regenerate multiple organs, and I think this is a key advantage over other animal models that are used, including mice.
So in adult stages, the zebrafish is not anymore transparent, but becomes like an opaque specimen. So we needed other different imaging techniques like the MRI and the microCT to try to understand how this structure is beating in a beating heart, for example with MRI, on how is the morphology of the structure more with the microCT. And the advantage of this was also that with the microCT we didn't need to extract the heart of the fish from the entire fish, and so we could actually image the outflow tract and also the connecting vessels of the fish without the need of destroying them or losing them during the dissection.
Performing MRI imaging on a beating zebrafish heart is quite a challenge, but the biggest challenge is the oxygen supply for the fish. So we have 2 possibilities. We can either perform it in a vessel, in a closed vessel with floating water through the vessel. Then the fish will have natural movement of the gills. Or we can put it without floating water, and then the gill movement will stop and we have only the heart movement. We still want to see the heart and the beating heart of the living animal. And so we use an intragate flash sequence, and this is a retrospective gating technique, and with this technique we don't need any external ECG. So this is, we can then detect with a navigator signal, we can detect the physiological functions of the zebrafish.
In the adult stages, many diseases regarding the heart can be modeled. For example, the cardiac hypertrophy, or another disease that can be modeled is the fibrosis of the heart or different cardiac arrhythmia. And also this can help also to understand better how the function of the heart restores after regeneration, so after myocardial infarction or many other different diseases. And for example, what we are trying to understand if zebrafish can also become a model to model diseases affecting the big arteries.